All right, well, welcome everyone. Thanks for joining us for 17 Minutes of Science. We might be up to the 18th program, but I keep losing track because we've been rolling for a while. So it's great to have you all with us. My name is Sarah Cheeseman. I'm a technical solution scientist at In Vivo Biosystems. And today we are looking forward to this conversation with my guest, Güneş Taylor, to explore a very interesting topic, which we agreed the title was Designing Better Babies. We can do it, but should we? So we're going to talk about that with Güneş here in a moment. Güneş is joining us today from London, so we appreciate her making time at the end of her day to join us more toward the beginning of our day. She is a postdoctoral training fellow at the Lovell-Badge Lab at the Francis Crick Institute. And this lab work revolves around stem cell biology and developmental genetics, and specifically, Güneş is studying methods of sex determination during development. And interestingly, she is also trained as a mental health first aider and is actively involved in science communication. So welcome, Güneş. Thank you for being here. And I would love for you to say a few more words about yourself.
Yeah, so thank you for inviting me. It was a real pleasure and honor. I'm really looking forward to this. This is something that I've mulled over a few times, but it's the gift that keeps on giving, as it were. So there's definitely more to talk about.
Well, that's great, because that's what we're here to do. So with that, let's go ahead and get a bit of a definition from you first about what is a designer baby.
Yes, absolutely. So technically, the phrase designer baby, I think, was first coined back in the 90s at some point. I certainly remember it being thrown around when I was at school. And it basically encapsulates any form of human being that has been modified using any means you like, theoretically, to introduce characteristics that you're interested in. So most people automatically think of the sort of aesthetic characteristics, so hair, color, eye color, stature, things like that. But it also theoretically encapsulates things like potentially mental capacity and general personality traits and things like that. So that was all part of the concept when the phrase designer baby first got coined. And yeah, it painted this picture where somebody could just go to a doctor and say, this is the kind of baby I want, and these are the characteristics, and somehow they'll just crank the lever around and this beautiful, perfect baby will pop out the other end like a Louis Vuitton bag, but better because it's yours. But, that's the idea anyway. That's the general thing.
Okay. I actually, yes, had to think for a minute about that connection to designer clothing, but that's a good one. That's funny. So, and it's important for us to remember that the term has been around for a while and certainly predates the technology that we're currently in, the CRISPR-Cas9 era, which is receiving a lot of attention, rightly so. And so perhaps you could tell us a bit about that technology as we continue forward in this conversation about what is CRISPR-Cas9.
So yes, genome editing is basically just changing the DNA of an organism using any means possible. And on the whole, a really good metaphor for thinking about this is in the following way, I think. So a genome is kind of like a complete set of instruction manuals, and imagine that the different pages contain different blocks of information and instruction on them, which can be thought of as genes, essentially. So old school methodologies would have been a bit like sort of ripping out pages of this instruction manual and shoving in other ones, and that's a form of genome editing. But CRISPR-Cas9 came along, 2014 was it, if I remember correctly, and it completely changed the way we edit genomes. And within the analogy, it's kind of a lot more like a find and replace function in Word for this sort of giant instruction manual that is your genome. So technically, CRISPR-Cas9, it has a two-part name. That's because it's a two-part system. The CRISPR is a small molecule that can be programmed by the user. That's the part where basically you say, find me this bit of DNA that I'm interested in. The Cas9 is actually a protein that cuts DNA, that breaks DNA. And basically, when you load these two bits together, it will go to whatever part of the genome matches that first CRISPR part, and then the Cas9 business end will break the DNA. And DNA is not meant to be broken. Cells have lots of mechanisms for fixing broken DNA, but with all mechanisms, mistakes can happen. And actually, in this case, when it comes to genome editing, that's exactly what we're after. So basically, you rain the Cas9 onto a particular site of interest, and the DNA will be repaired correctly many, many times, but eventually the mechanism will slip up and introduce an error of some description. And the idea is, hopefully, it's the type of error that would mean that we could, for example, break the gene of interest that we've just targeted, or so forth. So that's the basic concept. And I guess the last thing I should say about it is, actually it's come a long way in the sort of 5 or 6 years since this technology hit our shelves, as it were. And so now there's a lot more sophisticated approaches where you can, provide templates with which you wish to replace sections of DNA, and you can, tweak gene expression using modified versions of Cas9 that will, moderate the sort of general landscape around the gene of interest to make it slightly less on than on, or slightly less off than off, and things like that. So there's a lot more nuance to it nowadays.
So it sounds like the big advances have been around precision of using the tool for the editing event.
Yeah.
It's incredible to think that it's only been since 2014, because it's such a ubiquitous technology used across so many different kinds of animals and plants now.
Yeah, I mean, yeah, and that's because it's a spectacularly useful tool, regardless even from when it first was available and it wasn't even that, precise. Even then, it was more precise than anything else we've ever had before, and therefore even more tractable as a tool.
Mm-hmm. Which leads me to the next question about why is it that scientists are, despite this precision accuracy piece, still quite cautious about the technology?
Well, it's definitely a lot more precise than anything we've had before, but that doesn't mean it's the best it could be either. So, as I said, the whole premise is to just keep breaking a piece of DNA until you kind of get the outcome that you like, essentially. So there's an inbuilt bit of randomness there. And also, you have to consider that there are these things, I hate talking about it, but you have to bring in a bit of jargon. There are these things called off-target events. And if you sort of think about what that phrase means, it's exactly what it says on the tin. So off-target meaning, not the intended gene, or not the intended section of DNA has been targeted by the Cas9. So, for example, you could basically think you've had the editing event that you want because you're looking specifically in this part of the sort of instruction manual that you are focused on. But it turns out that phrase that you were targeting, that sort of section that you were targeting, might actually feature in a different chapter somewhere else that you're not even looking at. And so you might not even notice that that's also been changed. And that can have unintended consequences, theoretically. And this isn't just a theoretical thing, it's something that scientists have obviously were cautious about and have demonstrated do occur. So you always have to check for off-target events when you do any kind of CRISPR-Cas9 editing. And actually, very recent work from a group here in the Crick Institute actually has shown that this is also the case in human embryos. So this is not just, theoretically the case. It is in fact the case.
Well, that's a good segue, because I wanted to talk with you about the fact that, of course, this has occurred in human babies. And I'm specifically referencing the Chinese scientist He Jiankui, who several years ago made headlines when he announced, I believe at a meeting that your colleagues attended, as you told me before, that he had successfully edited human babies. So would love your thoughts on that. And then given that the children are several years old now, are we safe to assume at this point that it's been successful? And that sort of leads me into, this is now becoming a long-winded question, but thinking about the unintended consequences piece that you just brought up and how difficult it is to reckon with that, because we're talking about a lifetime of a person.
Yeah, that's the moneymaker right there, isn't it? Well, yes. So as you said, I'm actually very lucky to have a boss who is in such high standing in the world that he actually chaired Jiankui He at the conference over in Hong Kong, when the news broke. I think it was last January, actually, if I remember correctly. And so, yes, as you said, Jiankui He was a scientist. He was being a classical academic for many years. And then it was known that he was working on using CRISPR-Cas9 in human embryos, which in itself is fine. And then just at the last little bit, he sort of went a bit off the rails, as it were, and basically went underground and did his experiments and had them implanted into human mothers, basically waiting to conceive. And yes, it was shocking, disappointing, particularly shocking and disappointing from the perspective of the fact you'll remember that they were twins, and they were theoretically edited to target a gene called CCR5. It's the membrane protein, if I remember correctly. But basically, it's an important part of how HIV docks to cells and gains entry to them. And there is a naturally occurring mutation in certain populations on planet Earth that makes them slightly more resistant to HIV, and that's because they have a mutation in this gene. So these embryos were edited by Jiankui to carry this naturally occurring mutation, essentially. And one of the embryos had that said mutation, so the intended mutation was there. But the second one actually harbored a mutation that it was predicted bioinformatically, so using a computer to have a similar effect, but wasn't exactly the intended mutation in the first place. So that's sort of what happened. And talking about unintended effects, whether they were unintended or not considered by him, it is also known within the literature that having a mutated version of this particular protein, whilst it might be protective to HIV, it makes, at least in animal models, the carrier far more susceptible, particularly to West Nile virus, but I believe also other colds. So the theory goes, at least, whilst these babies should be protected theoretically against infection from HIV, they theoretically also are extremely vulnerable to catching things like a cold, basically.
Which arguably is a far more common measure than perhaps being exposed and infected, potentially infected by HIV.
I mean, the phrase that was used a lot at the time was an unmet clinical need, right? This was not addressing an unmet clinical need. So the fathers of these embryos were known to have HIV, but were under treatment regimes, which kept their viral loads very, very low. And so whilst, there is, on one hand, you might be able to imagine, one of the two parents is HIV positive, so maybe these babies would have been at higher risk than the population to being exposed to HIV. The fathers were actually under treatment, so they had fairly, fairly
Yes.
levels, right? So
It seems as though the concern of maternal transmission, should the mothers have been HIV positive, would have been a greater concern.
exactly.
So it's just very interesting. I think that aspect of the backdrop of this story was somehow lost in the media when they discussed it. And in fact, I don't know that I even knew that about the status of the fathers until you mentioned that to me. It was just not discussed about the pretext for why this was even done. So,
I mean, it has to be said that the most interesting thing about it was nobody really got to the bottom of, did he really think it was a good thing to do? And it seemed very much to those who spoke to him directly that he did genuinely think that this was going to be welcomed by everybody, that this is clearly a really good thing to have done. And I guess that's the moral of the story, isn't it? That what might seem perfectly reasonable, actually quite laudable to some people, can be received with unadulterated horror by others.
that is a good way to put it. And to think about how these kinds of advances in technology, there is such an ethical, societal component to it that really needs to be explored, because it feels like really transformational change.
Yeah, definitely.
So, yeah, we're right on the precipice of something, but we don't know what.
Exactly.
Well, we're in just approaching our last minute, and I know it goes like this, and I have more on that, but I would love to touch on another topic for a moment, which is your work as a mental health first aider, which we learned is something that your institution is investing in and supporting in. So if you could tell our viewers what that is, because I think for many of us here in the US, that is a novel idea of having the institution support that.
Yeah, absolutely. So yes, here at the Francis Crick Institute, there is a program to have a number of employees throughout the building on different floors in different types of roles, so not just scientists, trained to be able to act as mental health first aiders. And the idea is that we provide a support network which any one of our other colleagues can call upon should they find themselves in a very unstable or precarious situation in themselves at work, and really just need to talk to somebody, or really need some help pretty sharpish. And yeah, basically, so I've been trained to be able to engage with distressed colleagues and try and help mitigate those sorts of circumstances.
It feels like, given the moment that we're in, as this pandemic still continues to roll around the globe, and our lives are so affected, it feels like such an important thing that you're doing right now. And certainly on our shores here has elevated the discussion about the importance of mental health and to our whole selves. So that's great.
Absolutely.
Well, thank you. Our timer just made a noise, which means we've reached our 17 minutes, just like that. I want to thank you, Güneş, so much for taking your evening and spending time with us and talking about these varied and fascinating topics. We wish you all the best in your work. I hope that you continue to push the envelope.
Absolutely. Thank you so much for having me, and have a really great day. Yeah, right?
Yeah, lots of day left here. All right, well, thanks everybody for tuning in, and we will see you next time.